How do bacteria get antibiotic resistance through gene transfer? Imagine bacteria as tiny, clever organisms that can share secrets to survive. One of their tricks is passing on resistance to antibiotics, making infections harder to treat. This process is called gene transfer, and it happens in three main ways. The first way is transformation. Bacteria can grab free bits of DNA floating around in their environment. These bits often come from bacteria that have died and broken apart. If the DNA contains resistance genes, some bacteria can absorb it and include it in their own genetic material. This allows them to become resistant without needing to exchange DNA directly with another bacteria. The second method is conjugation. Think of it as bacteria shaking hands and passing a tiny circular piece of DNA called a plasmid. These plasmids often carry many resistance genes. When two bacteria come into contact, the plasmid can jump from one to the other. This transfer can happen quickly and even between different species of bacteria, spreading resistance widely. The third way is transduction. Here, viruses that infect bacteria, called bacteriophages, accidentally carry pieces of bacterial DNA, including resistance genes, from one bacterium to another. When these viruses infect a new bacteria, they insert the resistance genes into its DNA. Though less common than the other methods, transduction still plays a significant role in spreading resistance, especially in bacteria like Staphylococcus aureus. In addition to these processes, there are jumping genes called transposons. These pieces of DNA can move resistance genes within a bacterium's genome or between different DNA molecules. They act like genetic shuttles, helping resistance genes spread more rapidly and multiply within bacterial populations. For infectious disease specialists, understanding how bacteria transfer resistance genes is key to controlling antibiotic resistance. It helps them develop better strategies, like using antibiotics wisely and implementing infection control measures. This knowledge also guides research into new treatments that can outsmart resistant bacteria. So, bacteria share resistance through transformation, conjugation, and transduction, with help from mobile genetic elements like plasmids and transposons. This rapid sharing makes fighting infections more challenging but also highlights the importance of careful antibiotic use and ongoing research.